Alright, here we go. We're looking at 12.2 and 12.3, talking about linked genes and gene mapping. So we're looking at making a map of the order of genes on a chromosome. So here's a chromosome. We have gene A here, gene B here, gene C here, without physically looking at the chromosome. This is pretty cool stuff. And this is what's considered genetic analysis. So if you take genetics in college at all, it's going to spend a lot of time about genetic analysis. So using statistics, probability, and math between the outcomes of a genetic cross and the expected outcomes to learn things about how genes are situated in chromosomes. It's really what geneticists do a lot these days. So a lot of genetics is math is genetic analysis. So first, we have the chromosomal basis of Mendels Laws here. So what that means is that if we have a generation of parents that have maybe two alleles, Y for one gene and R for another gene, when they fertilize an egg, so both of the sperm and the egg gametes come together, their offspring will have any of these potential genotypes. They might have a dominant Y, a dominant R, a lowercase y, a lowercase r, a dominant Y, a recessive R, or a dominant R or a recessive Y. Alright? So, the reason that Mendel got those nice Punnett square numbers is because there's a 50-50 chance of which of the two homologous chromosomes is going to make it into a gamete. And then when those two gametes come together, there's 1, 2, 3, 4 possible different alleles that can be had in theirs. These 2, 4, 6, 8 arrangements of those alleles. So Mendel's laws have to do with meiosis, and that homologous chromosomes separate in meiosis 1, producing two uniquely different sets of gametes, not assuming any crossing over from one parent, and two uniquely separate set of gametes from the other parents. So the big thing we're talking about today is a linked gene, so alleles on the same chromosome. So genes that are linked, we say they're linked, are located close together on the same chromosome. So if these are both chromosome 1, both this gene, let's say A, and this gene B are on chromosome 1. That means they're linked. Anytime an individual inherits this chromosome 1, they will get both the dominant A and the dominant B allele. They are closely linked because these two genes are close together on this same chromosome. These two genes are less closely linked. I call this let's call this recessive A and recessive B. So, for example, if this was a pair of homologous chromosomes in meiosis, they'd separate. We'd have one gamete that'd have a chromosome with dominant A and dominant B on it, one chromosome with recessive A, recessive B, and a gamete. So you can either inherit a dominant A and a dominant B together, or a recessive A, a recessive B together. So it's more likely to have them both dominant A and dominant B, and it's very less likely to have a dominant A and a recessive B, because here those alleles are on separate chromosomes. It means that offspring are much more likely to inherit a certain combination, all dominant or all recessive, than for the other two genes, so one dominant and one recessive. That's why linked genes are important. They say something about the probability of inheriting different alleles together. Some genes are linked on our sex chromosomes, or X and our Y chromosomes. This means they're only located on these chromosomes. For example, the gene for color blindness is on our X chromosome. It's not on our Y chromosome. So it's linked to the X chromosome. There are some genes that are only on the Y chromosome. That means they aren't on the X chromosome at all. So, sex-linked genes is another name for genes that are linked to the X or Y chromosomes. This is because their presence is greater in certain genders. For example, if a gene is linked to the Y chromosome, that means only males could possibly inherit that gene. So, for example, if there is a disorder that only affects males, or is only on the Y chromosome, only males will be affected by it. Women don't have a Y chromosome, so they can't inherit that trait. On the other hand, if a gene is linked to the X chromosome, some interesting things can happen. One, if it is a recessively inherited trait and linked to the X chromosome, males will have a greater chance of inheriting this trait because they only have one copy of the X chromosome. So it's recessive. Normally you need 2 copies of that allele to see it. Otherwise it would be masked by the dominant allele. However, males only receive 1 copy of that X chromosome. So any recessive allele they get will be expressed. They don't have any allele on their Y chromosome. So it's much more common for a male to have an X-linked genetic disorder than a female. Here we have an example in a Punnett square. So let's say H equals normal vision and little h is colorblind. And we have a normal vision female and a normal vision male that are going to have children. This female is a carrier, so she has a normal copy of the allele and a recessive copy. Since it's recessive, she would need 2 recessive copies to have the disorder. Here the male has 1 dominant copy, he's not affected. But let's look at the offspring. None of the females on this side will be colorblind, guaranteed, because they will get 1 X from their father, 1 X from their mother, and the 1 X from their father is always going to have the dominant allele, so they will always have normal vision. However, if it is a male, there's a 50-50 chance they will be colorblind. If they receive the copy of the X chromosome with the normal dominant allele, they will be normal vision. They will receive the Y chromosome from their father. If they receive the recessive H from their mother and the Y from their father, they will be colorblind. Alright? That's why males, a lot more common to have X-linked genetic disorders in males. They only need 1 copy instead of 2. Pedigree of an X-linked recessive disorder would look like this. You'll see many more males are affected, and there's many female carriers. Here we only have 1 female that's affected, all these female carriers, but predominantly more women than men have the disorder. What if it's an X-linked dominant trait? If it's an X-linked dominant trait, then we can't really say that women or men will have it more often, because either gender will have the disorder if they only have 1 copy. But we can say something. We can say that an X-linked dominant disorder out of pedigree, if a male is affected, so too will their mother. Why can we say that? Because if there is a male offspring, the only chromosome as far as the sex chromosomes they got was their Y chromosome. That wouldn't have the allele. Which means that if they have the disorder, they received a dominant copy of their X from their mother. And if their mother had a dominant X copy, she must have been affected too. So, if there is a male affected, his mother has to have been affected for it to possibly be X-linked dominant. So this is something that's pretty cool with X-linked traits. Some things called X inactivation. So you might have wondered, females have 2 X chromosomes, males have only 1. How can that work? All genes make proteins. How can women have twice as many proteins as men? Well, you know what's cool? One of the chromosomes, X chromosomes in female cells, get deactivated. And it's called a bar body. It's an inactive X chromosome. So then both men and women have 1 active X chromosome. The fact is what X chromosome is inactivated happens very early in life, when there's only about 32 cells in your body. And it happens pretty randomly, so 50-50 chance. So women, in your cells right now, in your bodies, about half of your body has 1 X chromosome deactivated, the other has about the other X chromosome deactivated. Pretty weird, right? And you can see this in tortoiseshell cats. When they were young, one of the X chromosomes was deactivated, or the other. And when they only had maybe 32 cells big, when they were only like 32 cells big, and X inactivation occurred, maybe in 16 of them, this X chromosome that had the allele for orange color was deactivated. And the other half, maybe this chromosome with the allele for black was deactivated. And these cells multiplied and duplicated until we had an adult cat where some of their cells had the orange color still present, where the black got deactivated, and the X chromosome, and the other half of their cells had the X chromosome with a black color present, orange deactivated. And you can see where those cells migrated to around their body, because you can see the different patterning of where orange and black is. That's why the black and the orange will be in clumps, because those cells divided together. They came from common parents. Pretty awesome. The last thing we're going to talk about is recombination of linked genes and making gene maps. So sometimes linked genes can come unlinked. If the genes are on the same chromosome, they can end up switching to opposite chromosomes. This happens in crossing over, in meiosis, where pieces of chromosome break off and switch with each other. In this case, offspring may inherit a combination of 2 genes that were linked in their parents, but now are on different chromosomes. These are called recombinants. So for example, if this is metaphase 1 and meiosis 1, and A and B are on the same homologous chromosome, dominant A and dominant B, and recessive A and recessive B are on the same chromosome, you'd expect that the offspring would either have A, B, or recessive A, recessive B. But if these chromosomes cross over and this B, recessive B from this chromosome switches to this, we could get gametes that look like dominant A, recessive B, or recessive A, dominant B. These are called recombinants, because they recombine the order of alleles from the parents. Recombinants do not happen as frequently, so they're always the least common genotypes amongst the offspring, okay? Always the least common. So let's take a look. Using linked genes to make a gene map. Genes that are linked can separate through crossing over. Genes that are closer together on a chromosome will separate less frequently than those that are farther apart. So if we have a chromosome, genes that are close together, more likely, or close together, less likely to cross over. Genes that are far apart, more likely to recombine from crossing over. The percentage of recombinants from total offspring can tell us how far apart genes are on a chromosome, or if they are linked at all. So, if we would do this cross 1,000 times, and we would get a bunch of offspring, maybe 800 offspring that were either dominant A, dominant B, or recessive A, recessive B, and 100 that are dominant A and recessive B, and only 100 that are recessive A, dominant B. That'd be pretty strong evidence that they were linked, because there's far fewer...
If there's a DNA class in the building, please call 4405. DNA class in the building, please call 4405.
Far fewer recombinants than parental genotypes. So, for example, in this fly, B plus and VG plus are on the same chromosome. B and VG are on a different chromosome. If we cross these two, we can get a measure of their progeny. So we're going to cross a fruit fly that has dominant B, dominant VG on one chromosome, dominant B, recessive VG on the other. And we're going to cross another recessive fly that has 2 copies of the recessive gene. Okay? What we would expect is that just as many would inherit the BG plus and VG plus from the mother as B and VG from the mother, and that all offspring would inherit the lowercase b and lowercase vg from the father. But what we get out is that most of them have the 2 dominants or the 2 recessives, but some have one of both. We would expect all 2,000 offspring to have these if no recombination occurred. But recombination did occur. Some ended up with a dominant B plus a recessive VG. Some ended up with a recessive B, a dominant VG plus. So these are the recombinants. In total there are 391 recombinants out of a total of 2,300 offspring. If we just divide that and get a percentage, we get 17%. 17% recombinant. We have a term called map unit. That's just an artificial distance between 2 genes on a chromosome. 1 map unit equals 1% recombination. So this is 17% recombination. We can say these 2 genes, B plus and VG plus, are 17 map units away from each other. If we had a higher recombination rate, so 30%, that means that they're farther apart. It's more likely recombination is going to occur. If that was smaller, like 5%, that means less likely recombination is going to occur. So what is the distance between these genes? Let's say we have 2 parents. Parent 1 has the phenotype of dominant A, dominant B, and the second has a recessive A, recessive B. And they have offspring. So the possible gametes I could have on one chromosome, and on the other chromosome they're recessive A, recessive B. In this individual they have recessive A, recessive B. We're going to cross them. So dominant A and dominant B are linked, recessive A and recessive B are linked. They have 2,000 offspring. Of the 2,000 offspring, let's see, 800 are dominant A, dominant B. 800 are recessive A, recessive B. 200 are dominant A, recessive B, and 200 are recessive A, dominant B. These follow the linkage pattern, are what we call parental genotypes. They have the same genotype as one of their parents. We call them parental. these are the recombinants. Crossing over has occurred. Okay? Crossing over has occurred. So we're going to look at the number of recombinants. So in total we have 2,000 in total. And the recombinants here we have 400. Okay? So we have 400 divided by 2,000. And that is going to equal 0.28 or 20%. What we can say is that these two, A and B, are 20 map units away from each other. 20 map units. That's the distance between these two genes on the chromosome. If at all we get anything, a recombination frequency, so this percentage, if it's ever greater than 50%, it's not linked. Alright? If it's ever greater than 50%, our recombination frequency, it's not going to be linked. Alright, so here's just a look at what we're going to do on Thursday. We're going to learn how to predict whether a gene is linked or not using chi-squared, so I'm going to talk through chi-squared. And we're going to learn how to create a gene map with 3+ genes.